My name is Zequan Yang from the University of Virginia in Charlottesville, Virginia, United States. I am an associate professor of surgery and surgical critical care. I just presented my research at the International Academy of Cardiology annual scientific session in 2017 held in Vancouver, Canada. My title of my talk was New Insights into Myocardial Ischemia Reperfusion Injury, how to dampen a raging heart. Basically, I was talking about the cardiosplenic axis in mediating inflammatory response during ischemia reperfusion injury. So basically, my research has been focused on myocardial protection by inhibiting inflammatory response during ischemia reperfusion injury. So we have employed an in vivo mouse model with myocardial ischemia reperfusion injury by ligating left coronary artery. We also used several different genetically engineered mouse in several of my projects. The main theme of my study is try to define how the inflammatory response during ischemia reperfusion injury is triggered and how this inflammatory response mediates myocardial post-ischemia reperfusion injury. So we found this post-ischemia reperfusion injury is mediated by inflammatory response by activating a cardiosplenic axis. This cardiosplenic axis is triggered by ischemic rendered myocardial necrosis. Simply, ischemia rendered myocardial necrosis release damage-associated molecule patterns, including HMGB1 and the cell-free DNA, into the bloodstream. This molecule complex bind to the receptor for age on the splenic leukocytes. The activated splenic leukocytes was then released into the bloodstream, then migrate to the prior ischemic myocardium, homing there, causing infarct expansion during reperfusion. By inhibiting or depleting this HMGB1 or cell-free DNA can decrease the infarct size during reperfusion injury. So my results has several potential applications for clinical use. First, we found the ischemia in rendered myocardial necrosis is a prerequisite for reperfusion injury. There would be less reperfusion injury if the ischemic myocardial necrosis is smaller. So early restoration of blood supply to the ischemic myocardium is always the key to shorten the ischemic period, thus reduce the ischemia-induced myocardial necrosis and then reduce the reperfusion injury. Second, depletion or inhibit HMGB1 or depletion of cell-free DNA will suffice enough to reduce reperfusion injury. Third, control the concurrent inflammatory response induced by injury to other organs, like to the kidney, to the lung, to the liver, or the inflammatory response induced by hyperglycemia and sepsis may reduce the plasma level of HMGB1 or CF, cell-free DNA, and thus will reduce the inflammatory response systemically and have the potential to reduce the myocardial ischemia injury